Dear students, today I am going to start the topic cloning vectors, and I am going to discuss about the plasmids and pBR322. So, genetic engineering involves gene cloning, production of multiple copies of desired DNA or gene of interest. Now, the question is, what is cloning? The very word, the clone, K-L-O-N, it is the Greek originated, that means a twig. So, clone in a biology, a collection of molecules. So, you will find the whatever these are the plasmids inserted with our desired gene, they are multiplied in the test tube, or else they are multiplied in the culture medium. So, same many DNA molecules. So, this is a clone. Similarly, the clone is the cells, they are originated from a single cell and they are all alike. Genetically they are alike, and they are similarly plantlets you will find. Similarly, identical hexaplets. So, all these are clone. So, they have all are identical, have same genetic makeup. So, DNA cloning or gene cloning is a technique of reproducing DNAs, producing more DNA, same molecules will be. So, one DNA, the molecule, the same will be multiplied for millions or billions molecules are produced. So, that is what you can see, the gene cloning. The production of exact copies or the clones of a particular gene or DNA using genetic engineering techniques. DNA or gene cloning technique is to reproduce the DNA or gene into many copies. There are two methods. The one is polymerase chain reaction or PCR amplification. Then another is cell-based cloning. Cell-based cloning requires cloning vectors. So, these are required to carry the gene of interest or gene of DNA fragment from into the host cell for multiplication. So, that is what we are going to study today. So, we are going to study PCR technique later. So, today we are going to study the cell-based cloning, where you can see a molecular vehicle or cloning vector is required to insert the gene of interest, and that later it will be transformed into the host cell, which the host cell when it multiplies our gene of interest into many copies. So, this is what you can see, the cloning vector. So, the cloning vectors, gene cloning is to get a desired gene or gene product in large quantity. For example, it may be, for example, the insulin gene is inserted into the vector, cloning vector, then it is inserted into the prokaryote or bacterium, and the bacterium will multiply and it produces insulin in a bioreactor, and that can be separated or purified, and it is this is how exactly. Or else we can make many copies of this plasmid with the help of this bacteria, and we can able to collect and store it. So, these are the methods you can see. So, what is the PCR amplification is an in vitro amplification of the gene of interest, but there is a limitations. So, you will find the DNA, it is below 5 kilo base pairs, and that means 5000 base pairs size, it can be amplified with the help of PCR. The size, the gene size, if it is more than 5000 base pairs, it cannot be amplified with the PCR, it is not proper. So, that is why usually the genes, eukaryotic genes, they are the size will be more than 10 kilo base pairs. So, that is why we require cell-based cloning using cloning vectors. So, PCR in vitro multiplication, billions of copies like this, you can see this is a thermal cycler, and we can keep our DNA and we can multiply if it is 5000 base pairs less. So, now coming to the cloning vector, they are also called molecular vehicles or gene taxi. So, gene taxi or molecular vehicle. So these are the cell based cloning, where you can see gene of interest requires a molecular vector or cloning vector to carry the DNA fragment of interest into the host cell for DNA multiplication. Before cloning, DNA fragment must be inserted into the cloning vector. So the cloning vector has to be cut with the help of restriction endonuclease. Then we have to insert our gene of interest with the help of ligase, so that this recombinant the cloning vector or DNA we can able to transfer into the host cell. Host cell will multiply and we can get desired number of copies. A cloning vector is a DNA molecule that has an origin of replication and is capable of replicating in a bacterial cell. So because the bacteria replicate very fast, the multiplication of bacteria within no time, so the single cell will be millions of cells will be formed so fast. So that's why we can get our number of copies at a very short period. So that's why we require a cloning vector in such a way it can be transferred into the bacterial cell, and bacterial cell will multiply without inserting the gene of interest into the bacterial chromosome. That is also very important. That's what self-replicating. Using cloning vector, long gene 20 kilo base pair up to 250 kilo base pair can be multiplied. So that is the advantage. For example, mammalian gene or the human gene, those are all more than 250 kilo base pairs. So using cloning vectors, we can able to multiply these genes into large numbers. So the cloning vector is a DNA molecule, remember, having the ability to accept foreign DNA and can replicate independently in the host cell. So in the host cell, it is not disturbing the host cell chromosome, it will replicate independently. So that is the advantage of this cloning vector. There are number of cloning vector. You are not studying all those. You are studying some sample vectors, plasmids, cosmids, lambda pages, viruses, yeast. So these are all the common gene cloning vectors. You may ask why we need so many vectors. So this is what you can see, the vehicles, what we use. So we use a car for different purpose, bus, then the boat or the plane. So you will find, depending on requirements, depending on the size of the gene, so we use different types of cloning vectors or molecular vehicles. These are all you will find. They are all circular DNA molecules. They have some different characteristics. You will find how exactly all these are vehicles, but they have special features, the same thing you will find. So depending on our requirement, we will use these molecular vehicles or cloning vectors. So what are the steps you will find? We have to isolate that you have studied already. These are the basics of recombinant DNA technology. We have to isolate the gene of interest with the help of restriction endonuclease, and DNA sequence of the interest isolate from the rest of the chromosomes, so that we have to isolate. Then the gene cloning, gene transfer to the host cell through the suitable vector. So we have to collect the suitable vector, we have to cut the vector, we have to insert our gene of interest. Now this is a recombinant cloning vector. This recombinant cloning vector we are sending, or we are make this bacterium to accept this DNA. So this is the cloning vector. It is not going to disturb the bacterial chromosome. That's how the bacteria with this recombinant vector replicate, and within no time they produce thousands of the gene of interest. So we can lyse out this bacterial culture and we can extract this DNA that is in the plasmid, and we can also cut the plasmid and we can extract only our gene of interest, or else we can make extract the product of the gene of interest. This is how the cloning vectors will be helpful. So what are the general properties of a vector? Vector should be easily cut by restriction endonuclease. So it should have the restriction sites for restriction endonuclease. It should small manageable DNA easily transferable to any cell. So that is a small molecule, so it can be easily transported to the bacterial cell or any host cell. It should possess a suitable target site. The foreign gene of interest can be inserted without any disturbance to the functioning of the molecule. So it has a proper region, so where we can insert our gene, and it is not going to disturb this DNA as well as the host DNA. That is what you can see. The vector should be self-replicating, replicate independently without integrate into the host chromosome. Vector carrying a gene of interest should suitable marker genes so that they can be easily identified and isolated. Once we transfer, so we have to isolate. So when in this experiment, when we are transferring our gene of interest carrying cloning vectors, this transformation is not 100%, remember. So it is 10 to 20%. 10 to 20% of bacteria will take up our gene of interest or carrying cloning vectors, and rest of this are 80% they are not transformed. So we have to get rid of those things. So that's why we need a selectable marker. Then bacterial plasmid and other DNAs satisfy above conditions are engineered and developed for multi-purpose vectors. So now several vectors what I showed here, the different types of vectors, they are engineered vectors. They are originally taken from some prokaryotes or else some the cells, and they are engineered. So they are engineered means the required sequences of DNA will be attached. So the vector requires a multiple cloning site or a polylinker. So multiple cloning site, these are the general features of a vector. It should have a multiple cloning site or MCS or else polylinker. This is short sequence, this is about 20 to 100 base pairs, which contains you will find there are restriction sites for many restriction endonucleases. The restriction site for each endonuclease is different. So it is different for ECO R1 is different from ECO R2, different from HIND3, BAMH1, all those restriction endonucleases. So it should carry so that we can insert any this will be a multi-purpose. So this is what you can see. For example, a bus will carry the common people or else workers or else IT workers or else students. So any people will it is going to carry the same thing. You can see it should have it should accommodate any gene. So any gene means you can see the regular gene always they are having on each side there are restriction sites for a particular restriction enzymes. So these restriction enzyme sites, common restriction sites should be there in the multiple cloning site, that you will study more later. Okay. Then you can see the vector can be used for cloning varieties of DNA samples. So this is what you can see, this is a tractor. So this with the attachment it can do so many functions. It can it's for for transporting, it's for plowing, it's for watering, so many purposes, even for strike also it can be used. So this is what you can see similarly you can see the vector DNA molecule, it should have a multi-purpose value. So this many bacteria carry so we are going to study now cloning vectors as a plasmids. So what are plasmids? Many bacteria, they carry many copies of small circular DNA in addition to their genetic DNA or the chromosome. They are called plasmids or extra chromosomal DNA. You will find they are small molecules, circular molecules. You will see in the red. So this E. coli bacterium, it 3 plasmids. So sometimes it's to 5 to 6 circular DNA. They are of something like 2000 to 3000 base pairs of size. They are not, this is the real chromosome, that is the genetic material. So apart from that, it is if it is not there also, it is not make any difference for the bacterium. So it is carrying some extra chromosomal DNAs like that, you can see, those are called plasmids. Plasmids are self-replicating extra chromosomal circular DNA molecules found in most of the bacterial species and few eukaryotes also. So self-replicating means they have a origin of replication, they replicate independently with that of the chromosomal DNA, that's what you can see. So this engineered plasmids are extensively used for cloning vehicles in genetic engineering to perform many gene manipulations. This is how exactly we purchase a cloth and we can stitch into different types of we can make a bag out of that, we can use as a bed sheet, we can have a stitched into a dress, so many things you can see. So likewise the plasmid, here we extract the plasmids and then we can add many features, and this is called engineering. So this is what you can see, the engineered plasmids are extensively used for cloning vehicles in genetic engineering to perform many gene manipulations. So these the plasmids, they are taken from the bacteria, they cannot be used as vectors, it has to be engineered, it has to be modified, then only we can able to use for different genetic engineering manipulations. So the plasmids used in the genetic engineering have been artificially created, recombining fragments of various plasmids. So for example, some plasmids with so many plasmids from different bacteria can be clubbed and make one vector like that, you can see. What are the advantages of plasmid vectors? So engineered plasmid vectors will have a multiple cloning site. So it can be recognitions for many restriction endonucleases. So it is a restriction MCS or polylinker is a small 50 bases long DNA segment artificially synthesized, and it contains RNA recognition sites for more than 40 restriction endonucleases. So the plasmid vectors with inserted desired gene easily isolated from the host cell and transferred to the other cell. These are all the advantages of plasmid. So you will find this is a general plasmid. So any DNA fragment, this can be, in the case of any there are number of plasmid vectors developed now, so they are used, but there is a restriction, they can carry 6 to 10 kilo base pairs size gene easily inserted to this plasmid using restriction endonucleases and ligase enzyme. Then what exactly the multiple cloning site? This is what the general features of the plasmid. It is a origin of replication. It replicate independently without disturbing the genetic the chromosomal DNA. Then it is having a selectable marker. So this is coding for this gene is you can see beta lactamase gene. So it inactivates the ampicillin. It produces it's an ampicillin resistant gene. Okay. So this producing a protein which is bringing the resistance power to the bacteria carrying this gene. Okay. So this is MCS. This MCS having all these restriction site. So it has got a restriction site for EcoR1, SAC1, Capion1, SMAV1, BAMH1. So likewise SPA1, HIND3 like that you can see. So there are about 12 restriction enzyme cutting sites they are inserted artificially. So this is called polylinker. This is how exactly, this is a DNA, it has a sequence. So see this is the EcoR1 restriction site. This is next SAC1 restriction site, KPN1 restriction site, BAMH1 restriction, this is BAMH1 restriction site, this is SPA1 restriction site, the last, this is the HIND3 restriction site. So all along this you can see, this is what the sequence what you see, this is MCS, it is having the restriction sites for 12 restriction endonucleases, so that we can insert any gene of interest. Those can cut by EcoR1 or by SAC1 or by BAMH1. So likewise we have So that's why it is a multi-purpose. So today we study only pBR322, the plasmid. So this plasmid of E. coli developed by Bolivar and Rodriguez is one of the earliest plasmid vector. So that is PBR stands for plasmid. Bolivar, Rodriguez, they have developed, and 322, that is in their lab, it is the vector. They have given a numerical number 1, 2, 3 like that. The 322 is the number, in that you can see. So this is the widely used plasmid. It was prepared by 3 naturally occurring plasmid by removing unnecessary segments and adding essential sequences or restriction endonuclease recognition site. It is a selectable marker. So this is actually, you get confused. I will make a simple one. Next slide will show a simplified. So this vector is about 4.36 kilobase pairs. That means 4,361 base pairs. This entire the DNA, this the size. Okay, it has got more than 40 cloning sites. You can see. So pBR322 has a 2 selectable marker. So marker or antibiotic resistant gene. So you will find ampicillin resistant gene and tetracycline resistant gene. So the tetracycline resistant gene TET-R, ampicillin AMP-R resistance genes. R means R stands for resistance genes. They encode antibiotic resistance proteins. So you will find ampicillin antibiotic resistant protein is beta lactamase. So the gene coding, this is called BLA gene or ampicillin resistance gene. Okay, that confers resistance for ampicillin enzyme that destroys antibiotic penicillin class. Okay, so the DNA of interest can be easily inserted with these marker genes and selected. So the ORI represent origin of replication. So this point where the replication starts. ROP means that is the protein which restrict the replication of the plasmids. Then the insertion of the DNA at a specific restriction site. It has restriction site for EcoR1, HEND3. So likewise you can see the SAL1, BAMH1. So this is what you can see. This is a tetracycline resistant gene. If you insert your gene of interest in this site, so this tetracycline resistance property will be lost. So this has to be selected using ampicillin antibiotic. So this is called insertional inactivation. By inserting the gene, we are destroying or we are this gene is not going to be expressed. That is what you can see. Tetracycline resistance gene, by inserting a gene, the tetracycline resistant protein not going to be produced. So that has to be selected with the ampicillin, you can see. So that 2 types of selectable marker will be used. So how exactly they work? So this is our gene of interest inserted to the plasmid. It is carrying 2 selectable marker. We are inserting this into the E. coli bacteria. Those all this in a test tube, this type, so it has to be give a heat shock. Then only these bacteria will accept our the plasmid pBR322 carrying our gene of interest. Then we have to culture this in a plate. So what happens, this culture plate having a culture, it has got we have inserted our gene in the tetracycline with the help of BAMH1 in this region. Just imagine. So now we have to have a culture medium. It is carrying both tetracycline and ampicillin, or else it is carrying ampicillin. So what happens, you can see all the the bacteria expressing our plasmid will survive. Otherwise they don't have resistance power. If they have not incorporated with this plasmid, they will die. So these colonies are the one, they are having our the gene of interest, which is also having a gene of antibiotic resistance. So that's why they have colonized. You can see this white colonies, they are the transformed bacteria. or recombinant bacteria. So those who have not taken, because this recombination process is not 100%, it is 10 to 20%. So rest of the bacteria what we plated, they died. Only the bacteria, the recombinant bacteria carrying our gene of interest and also the plasmid with carrying the antibiotic resistance will survive. So this is how we can able to collect this, and we can transfer this into a large bioreactor, and we can get the product of the gene of interest, or else we can make it into multiple copies of this. So this is about pBR322 plasmid. Thank you.